Okay, thanks for coming. Thanks, Peter. I'm pleased to welcome a student from the Auckland Bioengineering Institute in New Zealand. Peter is currently on a research placement here in Chile, and he is working with Professor Julio Sotelo and Professor Carolina Saavedra to refine signal processing methods for endoscopic gastric bioelectric recordings. Peter from August will be based in the Traverso Lab at the Massachusetts Institute of Technology, refining device hardware using to obtain minimally invasive gastric bioelectric recordings. And today, Peter is going to present his talk titled Bioelectricity in the Gut, a new target for treating gastrointestinal disorders. So, Peter, you may begin with your presentation, please. Thanks.
Is there anywhere in particular to
Yes.
speak?
Yes, we have the microphones in this area.
Okay.
Yeah.
Amazing. Okay. Well, yes. Thank you for the introduction. My name is Peter. I am from Auckland, New Zealand, and here on the research placement, as you said, working with Carolina and Julio to refine the signal processing techniques for gastric bioelectric recordings. I'll present a little bit on that, as well as the sort of general gastrointestinal bioelectric foundations, and touch a little bit on the Auckland Bioengineering Institute, as well as to give a bit of context as to where I'm from and what we do. I'll also try and tone down the accent as much as I can. Yeah. From the Bioengineering Institute in New Zealand, we are what's called an LSRI for the University of Auckland, or a large-scale research institute. We sit alongside the faculty, so science engineering to large-scale research institutes at the Auckland University, sitting alongside those which don't run specific undergraduate programs. They're more research focused, although a bunch of institute members. We're about 300 to 400 people all up, a mixture of full-time researchers, lecturers, PhD students, master students, and administrative staff. And we focus on a whole range of different research areas. Our research areas are predominantly focused on medical technologies, or analysis and general research, or biologically inspired research more broadly, so things like biomimetics and the likes. Our structure is largely stratified into the different organ groups. So the brain, the guts, heart, lungs, musculoskeletal groups, with different niche topics spun off from those. But in general, we think of it as we have organ groups which focus on their areas of research. A few example projects that we have going on, just to give a bit of context. Example for the brain group, we're obviously a rugby nation, which involves a lot of concussions. So we do the likes of functional MRI modeling and analysis for all sorts of different populations. Populations. We have a lot to do with heart energetics and heart modeling, how it works in fighting different diseases in that respect. We do a lot of device development, which is what my area is focused on as well. But whereas I'm in the gut, we also have devices in the brain group, looking at hydrocephalus pressure monitoring. We have wireless power transfer, along with a bunch of other techniques in areas that we focus on. And for example, in the musculoskeletal group, we do a bunch of computational analysis in that area, looking at the effects of different surgical techniques on the structural abilities of the different bones and muscles after surgery, so that we can better inform surgical planning during surgery. There's a bunch of companies that. And then our flagship project that spans the whole institute is called the 12 Labors Project, which can be broadly defined as creating personalized digital twins for simulation of treatments for specific people. So it's taking all this research from all these different organ systems, combining it into a virtual model of specific people. So you can tailor treatment a lot better. For me specifically, I am part of the Target Lab, which translates or stands for the Translational Research in Gastroenterology and Emerging Technologies Lab. Of a mouthful, but it's essentially the GI lab. So we focus on the gastrointestinal system in all its different forms. So we look at different experimental and computational techniques for analyzing the system, how food moves, how nutrients are absorbed. We've got some device development going on in that. We have different patient data sets for nutrition effects that we look at with different machine learning techniques. But our primary focus is on GI electrophysiology. So understanding the electrical activity within the gut. And in its entirety, really, focusing on all the different organ systems, all the different organs within the gut. So from the esophagus down to the stomach. And we focus on that quite heavily, all the way through from benchtop testing and silico testing to human trials and commercial deployment at the other end. Okay, so we're going to dive into the gastrointestinal electrophysiology portion of our research a little bit more, because that is the main focus and it's where my particular research lies. So most people will know that the stomach has gastric contractions, which help move food and mix it and aid with digestion. But a lot of people will also not know that these contractions are governed and coordinated by an underlying electrical activity. So this electrical activity is generated by ICC cells in the gastric wall, or the interstitial cells of Cajal. And it's called slow waves. So these slow waves are ever present in the gut, and they trigger contractions and they entrain them in specific patterns that are established for healthy humans. These slow waves were first measured in the early 1900s by Professor Alvarez. And he did this using what's called an EGG. So a lot of people are familiar with an EKG, an ECG, essentially the same thing but for the gut, with sparse electrodes placed on the abdomen to record electrical activity in the gut from the body surface. Now, from these EGG recordings, you can obtain the frequency of the slow waves that move through the stomach. You can obtain an amplitude and a velocity. And this is useful in some degree to classify slow waves, but as it turned out, had limited clinical utility and limited ability to fully understand what was going on. So it kind of lagged in no man's land for nearly a century. Didn't see much clinical application because of the metrics that it could provide. Fast forward to the early 2000s to 2010, came the advent of high resolution mapping. So this was done through high resolution flexible arrays placed on the serosal surface of the stomach, so more invasive. But what it allowed us to do was get a spatial analysis of the electrical activity in the stomach, not just in one dimension. And this was pioneered by our group around that time. And the work that was carried out with high resolution mapping and these flexible arrays determined that there was a regular spatial pattern for slow wave propagation in healthy humans. So this, this activation starts in the area of the upper corpus of the stomach, which you should be able to see on the video. It's on the sort of mid-right. And then it spreads rapidly to form ring wavefronts around the stomach, which travel distally towards the pylorus, or the ends of the stomach. And you can have more than one wavefront traveling simultaneously.
Sorry, what is the first image?
This one?
Yeah.
So the image on the left is stacked flexible arrays. Okay. So that all these little points are electrodes.
Okay.
And it's on, I think it's a polyacrylamide flexible printed circuit. So
and you put that inside the...
Yeah, so the second image is an outside. So these are the ribbon cables coming off from that printed circuit to the acquisition hardware. And they're placed on the serosal surface of the stomach and conform to it. So there's generally 256 electrodes in that array to give a dense array to properly quantify in detail where the activation is moving. Yeah, you might question why this is actually important. Why do we need spatial awareness? What clinical utility does it give? Well, the breakthrough with this high resolution mapping and the spatial awareness that it gave was that in certain subsets of patients, it gave, they presented with spatial abnormalities at normal frequencies. So the activation that you would obtain through EGG recordings or simply velocity one-dimensional recordings, it would appear like it's a normal patient. But if you looked in more high resolution in the spatial domain, then you could see that it was either moving backwards or turning around at a certain point. So you could see there were actually abnormalities that you could tie to these specific patient groups. And that's important because it gives us a target. We can say, okay, these patients actually have something that we can then go and hopefully treat. And the specific diseases that these abnormalities were found in were generally functional disorders. So gastroparesis or functional dyspepsia, which have quite a significant effect on the population. So combined, they are considered to affect about 15% of the population, give or take wherever you get the data from, and over $20 billion in total combined effect on the US economy, at least given the last data that was collected on that front. And an interesting point to note with these spatial abnormalities that were identified was that they exhibited similar characteristics to dysrhythmias in electrical conduction observed in the heart. So there was reentrant activity where we're draining around a specific point. There was conduction blocks that would stop propagation. There was retrograde propagation in the wrong direction. It's interesting to note, and something that I will touch on in just a bit. One of the problems with the current treatment pipelines for these functional disorders at the moment is that they're incredibly poorly understood. So the underlying mechanisms haven't really been teased out before this, and the treatment pathway for them typically involved an exclusion diagnosis. So essentially you run through all your different tests for all your different diseases, get to the end and say, you don't know, you must be dysfunctional. There's still a bunch of sub-classification to do because of this. Essentially, you get to the end, like, I don't know, you must be this. And the treatment options available for them are just poor because of that lack of understanding. So throwing a bunch of stuff at the wall. A lot of them are pharmacological treatments, just targeting symptom relief for the life of the patient, or more invasive removal of muscle to aid in accelerating gastric emptying, moving the food through, because delayed moving the food. Hallmark of these diseases. So we also need some better options to target these diseases. Is looking at the similarity that the electrical propagation drew with cardiac dysrhythmias. There was sort of thought that is the treatment pathways that are typically used for treating these cardiac dysrhythmias would be a good place to start. And a standard procedure or treatment pipeline for cardiac procedures is ablation, which is essentially finding the dysrhythmic tissue and burning around it to isolate it and stop it interfering with the rest of the circuit, which is quickly normal. And this ablation treatment is incredibly successful in cardiac applications. So the promise of translating it to the gastric arena was quite large. The pipeline, in general speak, for cardiac ablation is using your EKG or ECG to identify which patients might have an abnormality in the most or in the least invasive way, so on the body surface, and then you would send in an endoscopic catheter to map the activation in much more detail, find exactly what tissue is misfiring, and then you would send in the ablation catheter to isolate that. So we've done the same for the gastric arena. So instead of an EGG, we've moved more towards what's called body surface gastric mapping, which is similar in principle, but more electrodes and a two-dimensional array, so you can get more information. So that is essentially the EKG step for the gut at this stage. And then my project is the endoscopic application, so designing an endoscopic catheter to go and map the activation in more detail. And then there is another part of the group that's working on the ablation side of it, sort of final step in that pipeline. Just I'll touch on the other two parts before diving into myelin, just to give a sense of where the pipeline is at. So for the body surface gastric mapping, we have a spin-out company from the group called Telemetry, which is FDA approved, operating in the States, UK, Australia, and New Zealand, I believe, led by Professor O'Grady, who pioneered a lot of the original serosal mapping as well. So that's in clinical deployment, targeting classification of patients and trying to guide treatment in that array. And then the gastric ablation work, we have a part of the group that is working on that, which is going really well. We have done acute porcine trials, which show that we can isolate dysrhythmias and resolve them and alter conduction properties through ablation. And we've also done some longer term chronic studies over 2 weeks, which show safety of the procedure at least over that 2-week time frame, and with no complications in that particular study. And they're gearing up for some more endoscopic delivery of the ablation catheter and refinement of the technique, as well as hopefully human trials in the next year or two. But the endoscopic part, so the middle part that my research is focused on. To facilitate the endoscopic mapping in more detail to try and guide that ablation treatment, we designed a device. It's about 60 mil in diameter. It's got 64 platinum iridium electrodes, equally spaced over 8 splines. So it's a repurposed Boston Scientific Constellation catheter with a balloon in the middle and spacing splines around the outside and a full attachment on the top. We have used this device to take recordings in a human trial in Auckland and Christchurch City Hospitals in New Zealand. The process of delivering the device is largely shown in these bottom images, with the device delivered alongside a gastroscope down the esophagus into the gastric pancreas, which is the second photo. And then once it's placed in the gastric pancreas, it's inflated to space out the electrodes evenly, and the stomach suction down onto those electrodes to maintain contact. And it's left there for about 7.5 minutes as part of the regular endoscopy procedure, after which we deflate it, remove it, and do all our post-processing afterwards. And we process in a custom software to give quantitative measures such as those velocity measures, but also spatial visualization. The custom software that we use is called Visoa. It was developed by a previous PhD student, Alex Chang, and it's tailored for the analysis of bioelectric recordings with a bunch of customization and visualization options, and it is readily available on GitHub for use if anyone wants to use it for any other application. So from the patients that we obtained recordings from, we obtained slow wave signals with frequency about 3 cycles a minute, about 5 millimeters per second velocity, and just over 2 millivolts amplitude, which all fit within the established physiological ranges for slow waves, demonstrating the validity of the device with no complications in any of the patients that we tested with. We have expanded this trial to 35 patients, but we're just running through some more analysis on additional patients as well. Interestingly, there was a split that was statistically significant between the SNR in patients which we were able to detect slow waves on greater than 33% of the channels on the device, and patients below this threshold, which we hopefully indicates that there is underlying slow wave signals in those recordings that we could extract with more aggressive filtering and processing, which we're currently investigating. Just in a brief conclusion and to give some context where we're going on this project, we have managed to record gastric slow waves in humans by minimally invasive endoscopic delivery for the first time. So this demonstrates that the technique is safe and feasible for potential clinical application, but that these recordings have exhibited significant noise, so we need to address that with additional post-processing and hardware requirements. One of the limitations that we found was the size of the device. So right now, the device is adapted from a cardiac device, so it's limited in size to what it could be. So we need to expand that out to cover the bottom 2/3 of the stomach, so the corpus and the antrum, which are the 2 electrically active parts, is what we will be targeting with the Traverso lab from August. And then the final step would be to integrate these three processes. So the closed spectrum mapping, the endoscopic mapping, and the ablation into a deployable pipeline. Hopefully help. I've just got a bunch of projects here as well in case anyone was interested for students. They're all funded. Yeah.
Thank you.
Yes, I have a question. You said that from the 25 subjects, you get recordings from 22.
Yep.
What happened with the other 3 subjects? Why you couldn't get the recording?
Yeah, so the other ones, they were just too much noise. So,
okay.
so like
it was a problem with the noise, not,
Yeah.
not with the device.
yeah, so the 25 patients, we obtained recordings, so the device was delivered successfully and retrieved, but we just couldn't pick out flow waves from it.
Okay, and all of them are patients. Do you have recordings with healthy subjects?
So they're all scheduled for endoscopy for some reason. So they're all coming in for treatment of some problem. So we can't piggyback on the back of that or resources too much. But there is subsets of those patients that you could consider electrically healthy. So yeah, so they've got other problems that you wouldn't normally associate with electrical abnormalities, like ceiling. So you could use those particular patients as electrical normal group, and keep the other ones that you have where you suspect functions. That's a little bit tough at times because often the diagnosis is made after that endoscopy that you're piggybacking on, but we have to jump in. Well, so you kind of have to make a guess, be like, oh, we think this patient will probably get this diagnosis, and go from there. So it's a little bit difficult to tailor the cohort exactly, but there is a potential to explore out those.
Very interesting work. I didn't know that you can measure the bioelectrical signal of the stomach. So, do you know if is there some research or something related to this and cancer?
It's not the focus of it, and I don't think there's been a whole lot gone into it. I'm not aware of any specific states targeting cancer, but off the top of my head, cancer does cause structural abnormalities, so the potential. Yeah, I assume this at the moment that the malignant nature of the more of a target than electrical abnormalities. Yeah, pretty interesting.
Yeah. Well, thanks again for your presentation. I have to say that there are people connected in the streaming. They don't have questions, but we have people watching your presentation. Okay, thanks. Muchas gracias.